Good morning. It's November 7th, and this is your daily brief in science. Here's everything you need to know. A reshaping of focus at the Zuckerberg initiative pivots toward Biohub and science, with artificial intelligence driving biomedical discovery and AI-driven virtual cell models to understand human biology and disease. Biohub plans to double its basic science funding over the next decade, targeting roughly a $1 billion annual operating budget after contributing about $4 billion since 2016. This move follows criticism of scaling back DEI and immigration-related funding and signals a long-horizon strategy to fund 10 to 15-year tool development in biology. The description outlines a high-level restructure and acquisition without detailing financial terms, dates, or organizational structures. Stakeholders expected to benefit include NVIDIA for hardware, CoreWeave for cloud, Vast Data for storage, cloud providers, Meta platforms, and specialized AI biotech startups with evolutionary scale acquired to bolster capabilities. Open science and large-scale data sharing are central to the strategy, potentially challenging proprietary AI ecosystems and pushing research toward collaboration and data-centric models. The restructuring is framed as a long-term bet on AI-enabled research infrastructure with implications for grant-making, partnerships, and collaboration with academic and industry researchers. Core technologies include multi-scale, multimodal virtual cell models like TranscriptFormer and GREMLN, supported by an HPC cluster with over 1,000 NVIDIA H100 GPUs and a managed Kubernetes environment. Initial scientific skepticism about such a grand mission is acknowledged, but proponents say new tools can enable breakthroughs by observing phenomena in novel ways. The initiative aims to invert traditional workflows by testing hypotheses computationally before lab work, potentially compressing years of research into days and improving discovery success rates. The move occurs against broader cuts in federal science funding and public health investment, underscoring a push toward long-term, high-impact science spanning a decade or more. A new study from Dalhousie University finds microplastics in Nova Scotia lobster tails, traced mainly to polyester clothing, industrial adhesives, and marine-grade materials, raising questions about lobster health and potential human exposure. Researchers used Nile red staining to visualize microplastics and Raman spectroscopy to identify plastic types, sampling 16 lobsters from 4 fishing areas that represented different sizes and sexes. Findings show microplastics can increase mortality, trigger inflammation, and cause cellular damage in crustaceans, potentially affecting nutrient uptake, oxygen use, and body mass in lobsters. The study highlights the broader context of Canada's plastic waste challenge, where nearly 5 million tons of plastic were produced in 2021 and only about 7% recycled, with most waste ending up in landfills or the environment. The work underscores a need for further research on how microplastics move through marine ecosystems and what this means for fisheries and food safety in eastern Canada. Meanwhile, Canada's budget signal points to significant net new spending over 5 years amid discussions of global disruption and rapid tech change, with implications for manufacturing, construction, and public services. In other developments, Indigenous leaders criticize funding gaps in healthcare and education within the federal budget, calling for generational investments to address housing and Inuit-specific needs. Nova Scotia MP Chris D'Entremont has joined the Liberal caucus, part of a broader effort to present a unified approach during economic uncertainty. The report also notes lobsters' economic importance to eastern Canada, arguing for focused study within the plastics pollution discourse while acknowledging the global prevalence of microplastics across marine species. A Nature metabolism study points to epigenetic clues in kids born to mothers with type 1 diabetes, suggesting DNA methylation changes in blood may help explain protection against islet autoimmunity. The researchers linked several differentially methylated CpG sites to gene activity, identifying 142 genes with expression correlations, including several type 1 diabetes susceptibility genes in the MHC region and HOXA genes, with 105 CpGs potentially regulating 15 type 1 diabetes-related genes. The work builds on observations that offspring of mothers with type 1 diabetes have lower risk than those with affected fathers or siblings, offering a mechanistic angle through epigenetic regulation. A cluster of CpGS maps to HOXA genes on chromosome 7p15, notably HOXA5, and promoter regions show methylation differences confirmed by bisulfite pyrosequencing. Compared with older offspring studies, exact CpG or DMR overlaps are limited, but several shared target genes point to similar pathways. Motif analysis highlights enrichment of MBD2 binding sites, a regulator of T-cell development and regulatory T-cell function, suggesting immune pathway involvement in the epigenetic changes. About 198 CpGS in offspring of mothers with type 1 diabetes associate with 239 circulating protein traits, including CRISP2, a biomarker linked to progression to type 1 diabetes. The discussion highlights the polygenic nature of type 1 diabetes with MHC locus primacy, possible roles for MBD2 and TREG regulation, and the HOXA or HOTAI-RM1 axis in immune development, proposing targets for functional validation. Methylation differences concentrate in promoters, 5' UTRs and CpG islands, with regulatory element enrichment across blood cells and tissues relevant to type 1 diabetes, such as pancreas and thymus. Further work is needed to define scope, significance, and practical implications for offspring health and development. A follow-up project, supported with over $550,000 from the Leona M. and Harry B. Helmsley Charitable Trust, will identify which susceptibility genes are epigenetically modulated by maternal type 1 diabetes, assess similar effects in gestational diabetes, and explore protein and metabolomic biomarkers across cohorts, GPPAD, Baby Diab, Baby Diet, Frida. The collaboration spans institutions across Europe and totals funding of $550,000 to quantify protective epigenetic effects and extend analyses to gestational diabetes and multi-omics. Methylation propensity scores, or MPS, were developed to capture maternal type 1 diabetes-associated methylation patterns, with 34 CpGS linked to type 1 diabetes susceptibility genes and 426 CpGS from non-susceptible loci, achieving AUCS of 0.71 and 0.88 respectively. Findings align with risk variation by parental status, with higher risk when fathers or siblings are affected, prompting exploration of maternal health effects during pregnancy. In offspring without maternal type 1 diabetes, higher MPS at type 1 diabetes susceptibility loci associates with reduced islet autoimmunity risk, odds ratio 0.57 per unit MPS, replicated in the Frida cohort. The study presents a preview of nature metabolism content, linking environmental and familial factors to autoimmune risk and grounding the work in foundational epigenetic research. The analysis drew on blood samples from 1,752 two-year-old children, comparing 790 with maternal type 1 diabetes to 962 without, all at elevated genetic risk for type 1 diabetes. Type 1 diabetes involves autoimmune destruction of pancreatic beta cells requiring insulin, underscoring the importance of understanding epigenetic influences on disease risk. A 7-day immersive mind-body retreat at UC San Diego spurred rapid multisystem biological changes in 20 healthy adults, with brain imaging and blood markers indicating neuroplasticity, metabolic shifts, immune activation, and pain relief mechanisms. The program blended meditation, reconceptualization, and open-label placebo elements, including guided practices and Kundalini techniques, producing broad neural and molecular effects that researchers describe as systemic and lasting beyond the retreat period. Post-intervention analyses found reduced connectivity between major brain networks and key nodes, a notable increase between the left insula and posterior cingulate linked to absorptive contemplative states, and exosome-derived non-coding RNA expression changes influenced by time point and practitioner experience. Metabolic profiling showed post-retreat plasma promoting higher glycolysis in neuron-like cells, indicating a more flexible metabolic state with altered mitochondrial respiration, while endogenous opioids rose in the blood, signaling activation of the body's natural pain relief systems even in an open-label context. Gene and small RNA analyses in blood pointed to shifts in brain-related pathways, and immune signaling became more complex with simultaneous rises in both inflammatory and anti-inflammatory markers, hinting at a nuanced recalibration of immune responses. Mystical experience. Questionnaire scores increased after the retreat, with higher scores correlating with greater brain network integration and broader biological changes, suggesting a link between subjective experience and objective molecular and neural shifts. The study, published in Communications Biology and funded by the Inner Science Research Fund and VA Research Career Scientist Award, notes an author's employment by Encephalon Inc. and calls for longitudinal studies to assess durability and whether repeated retreats amplify or stabilize these effects, including potential benefits for chronic pain, mood dysregulation, or immune dysfunction. MIT researchers present clear evidence of unconventional superconductivity in magic-angle twisted trilayer graphene using a novel platform that couples tunneling and transport to observe the gap in real time as superconductivity emerges. The setup merges electron tunneling with electrical transport, enabling the gap to be identified only when zero resistance is achieved. The study reports a sharp, V-shaped superconducting gap in MATTG, marking a direct signature of superconductivity in this material. Funding support came from several U.S. agencies and foundations, including the Army Research Office, the Air Force Office of Scientific Research, the NSF, and the Gordon and Betty Moore Foundation. The science paper is led by Pablo Jarillo Herrero with Xuen Sun and Jongmin Park as co-leads and includes collaborators from Japan's National Institute for Materials Science. Co-lead authors include Sun and Park, with collaboration from Kenji Watanabe and Takashi Taniguchi of NIMS, highlighting a joint MIT-NIMS effort. The article is titled Experimental Evidence for Nodal Superconducting Gap in Moiré Graphene, published November 6, 2025, noting funding from multiple U.S. agencies and foundations. The broader context sits in the field of twistronics and the ongoing pursuit of room-temperature superconductivity. The findings suggest electrons in MATTG pair through electronic interactions rather than lattice vibrations, indicating a potential path toward higher temperature superconductivity through material design. These observations imply a pairing mechanism driven by strong electronic interactions and point to a distinctive symmetry in this material. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.